I'm not sure what constitutes a quorum for this group just yet, but I'm going into our 8th hour.
Yeah. I know, and it's only day one.
The beauty is I'm not presenting tomorrow.
Yes.
It's you guys, it's going to be your turn.
Yeah, it's going to be our turn tomorrow.
Yeah. So I will probably need some input from you guys to keep my energy up, since it's I feel like I'm in this weird Zoom space where it's actually it's supposed to be 3 o'clock in the afternoon, but it feels like it's evening, and it actually is evening. At least it's not getting dark.
Yeah.
But I'm going to hold on to that last sip of water because I might need it.
I think it's still better than midnight in Europe.
Yeah, it's definitely better than midnight in Europe or other. I don't know where other places other people are at this point. We had people in the last workshop all over. I mean, we had some in India and in other 11 p.m., 1 a.m. here.
Yeah, last time. There we go.
Wow. Well,
last year we had someone from Nigeria, someone from Australia. Australia was the most distant time zone, I think.
Yeah. Sometime we should target, when we get better at this, we should target some workshops in places like, like one for Oceania or something, because I'm sure that they would be, appreciated, and it would be fun as well. But it would take some careful planning on our part, or at least some sleep deprivation. But it'd be fun to try that.
Yeah, Terry travels the world so much. He's got a recipe for how to change your time zone.
Mm-hmm.
But I had an interesting experience a few years back when I traveled to India to visit with Suhita in Pune, and that was my first time traveling there. And I think it was like 23 or 24 hours in the air on the airplane, and I arrived in New Delhi at like 8 in the morning. And they say to try to stay up till local bedtime, right, before you go to sleep. Of course, it was 8 in the morning, right? So I tried. But the funny thing is, ever since that trip, I no longer get jet lag. And I talked to Terry about this, and he thought maybe I'd done some brain damage or something, but I think he was just joking with me. But really, I don't feel like I get jet lag anymore when I travel places. And it happened right after that trip. I don't know. Anyway.
Yeah. Well, we'll see what happens when you start traveling internationally again.
Yeah. Yeah, exactly.
Hopefully you won't get jet lag. That would be nice. That's definitely nice not to get jet lag. All right. Like I said, so keep the comments coming. The more interactive this is, the better I'm going to be able to provide interesting content. I'll just say that. I wanted to go back to the last thing we were talking about, and I added a few slides to my presentation, and I pushed my changes. Although I have to say that I'm still getting used to interacting with Git, especially in VS Code. When you run so if you run BioNetGen and you do things like parameter scans and bifurcation under revision control, under directories that are under revision control, it generates huge numbers of changes that you have to put in git ignore somehow, and I haven't figured out efficient ways to manage all of that yet. So I'm still struggling, but I did manage to push just the PowerPoint files to the repo, so I was happy about that. let me share my screen before I can pull up the presentation. So we stopped more or less here. We did this parameter scan. We didn't, this is before the bifurcation diagram in the notes, but the key idea to understand what's going on is that if you make a plot of the production rates and the degradation rate, so the production rate is the rate at which K1 P or the phosphorylated kinase is formed, you can with a few assumptions you can do this analytically and make a plot, and it looks more or less like a parabola as a function of the AP concentration. It's this red curve, and the degradation rate, so due to the phosphatase, the dephosphorylation rate is this blue curve. And remember, I said that you have to be operating at saturation in order to observe the bistability, and that's because this curve has to bend in order to get multiple fixed points out of this. If you just have a straight line dephosphorylation rate, you're just going to get one intersection between the curves, possibly two if they both start at 0, but you won't get the 3 that you need to have a bistable system. So we actually, I did some, this model has been analyzed in more detail. And one place that you can find that analysis is in this paper that we put out last year. That was actually, we were interested in looking at basically space and stochasticity in cell biology, in simulations of cell biology and cell biochemistry. And it was a fun working group that actually grew out of Bios, Bob Murphy, whom you'll meet shortly. And I organized this working group that was organized out of the University of Tennessee. They had a center for, I can't remember what the acronym means. It's NIMBUS is the acronym, NIMBIOS. And we met together for a while, over the period of a number of years, and developed several benchmark models for testing the validity of different spatial simulation algorithms. And one of the benchmark models was this autophosphorylation circuit, just to demonstrate the effects of bistability. It turns out it doesn't have a lot of space. This model doesn't have a lot of spatial effects that really are easily accessible within biological regimes, but it's a pretty cool model from just a nonlinear stability point of view. And we already saw the effect of the bifurcation scan. And a fun exercise with this model. So I mentioned before that stochastic effects, and there's a nice example of stochastic effects in the population model at the end of the lecture notes from the previous section. But those the stochastic effects that occur there have to do with the fact that when you have discrete populations, you can have quenching effects where the number of individuals goes to 0, and then things can shut down. Processes in your system can shut down, and it's much harder to do that with continuum systems. So you can see some pretty radical switching behaviors and very strong departures from the average behavior in stochastic trajectories due to these kinds of quenching effects. But bistability is another area where you can see really strong departures from the mean trajectory behavior in from ODEs. And so if you, as an exercise, run a long stochastic simulation of this autophosphorylation model. So I put this as an exercise to describe, to do this and describe what you observe. So we had, if you uncomment let's see if this SSA command here. I mean, what the heck? We could just do it. Why is this? I'm wondering if I haven't done this one before. but I think this is the one I'd use for bifurcate, but maybe not. Oh, maybe, no, I did it here. I have multiple repos open for different reasons. Okay, so I have to bifurcate, so I can comment that. If I come back to this SSA and run this, I think it'll show something very similar to what we had in the paper. You can see it runs very fast. It may, the plotting, oh, this one isn't very exciting. Oh, probably because my initial conditions aren't right. So this one doesn't switch. I forgot what I have to do off the top of my head to make it switch. Just trying to remember. I don't. I wonder if I have to set. No, not working. There is a way to make this thing behave like so. Probably because yeah, that's an example. The S equals 0 behavior is an example of punching. If all the kinase is unphosphorylated and you don't have this a mechanism for spontaneous phosphorylation to occur, nothing will happen. So maybe I'm really just guessing. I should stop, but maybe this one will do it. Now it broke. No, didn't work. Well, if you get the parameters right, the conditions right, what you will see is what's shown in the slides as a solution. So you can see trajectories that look like this. So the blue in these trajectories is the phosphorylated form of the kinase. Here, the kinase is called A instead of K, but okay. And so you can see these, what happens is you'll be in one regime in the upper state, and then it'll switch to the lower state. Actually, it's reversed. This is the lower state, and this is the upper state. And you can switch back and forth at more or less, random intervals between hops, and it stays in the upper state considerably. The upper state is more stable. It stays in the upper state longer than in the lower state for the parameter values that were chosen in this simulation. And the values are actually reported in the paper, but the way that model is formulated is slightly different than the way ours was formulated. So if people are really curious, I can look up I can get the parameters that give rise to this kind of nice switching behavior when you look at a stochastic trajectory. Should be easy to figure it out. I just am drawing a blank off the top of my head exactly what I have to do. It's very sensitive to the, people might have said something in the chat here. Okay. It's very sensitive to the number of phosphatase molecules, again, because of that saturation effect. So if you don't get that right, you won't see what in the world happened here. Oh, this is the wrong file. Yeah, it might be something as simple as lowering this to, it's like 108 and 9 or something that you have to set it to. So it can be very tricky to get this point just right, to get it to switch regularly. But anyway, so that's a bistable switch. So it's a fun model to play around with, and again, it exhibits some both nonlinear behaviors that are very strong and stochastic behaviors as a result that are very strong. And let us then, if there are no questions about that. Oh, I see one new message. The suggestion here is to increase S. Well, I'll let you guys play with that because I know I'll just, yeah, I don't want to get too lost in that for the moment. We can come back to it later on. Okay, so one thing that we skipped over before, and I promise, I feel obligated, I promise to talk about compartmental models a little bit, because it's important for the linkage between BioNetGen and MCell. And it's worth trying to cover now, even though the time is late, and it's really late for some of you, because the repetition will help. So if you see it a couple of times, it'll sink in better. So how do we? So the idea is that we want to make models that have the spatial topology defined in them. And what does that look like? So so far, we've basically assumed that all of our reactions are occurring in a single compartment. And in fact, we're not actually doing that when we model things like ligand receptor binding, but we're kind of accounting for the compartments in a sort of an implicit way. And that's not going to work very well when we want to make spatially explicit models. And so to be able to more easily transition between defining models as reaction networks and defining models as explicitly spatial, where molecules are located in particular regions of space and can react with each other, we have this. We can segregate space up into compartments, which are really topological regions that are connected to each other, but don't have an explicit geometry necessarily associated with them. So what do we mean by that? So if we think about a typical cell, it's bounded by a plasma membrane that represents the surface of the outside surface of the cell, and that borders on the space that's surrounding it. And we've got PowerPoint jumping ahead again. What's great is that it'll jump ahead from my mouse, but when I go to the keyboard, it starts barking at me because I don't have the window selected, so I don't understand that. But okay, the plasma membrane is here, the extracellular space is here. So here's our cell. There are molecules diffusing outside the cell that can bind to things that are on the surface of the cell, surface receptors, for example. And then when that binding occurs, the receptors can communicate in some way that the binding has occurred through various mechanisms, either through a conformational change or by associating with other surface receptors and forming complexes. And really, that's the sort of one of the basic origins of the combinatorial complexity is this need for the surface receptors to aggregate. And then molecules that are inside the cell can associate with those receptors and nucleate signaling complexes, right? Ligand-gated ion channels too, definitely. And they often, the ion channels often associate with the receptors and so forth. But so information about binding events that are taking place outside gets transmitted inside through the aggregation of complexes, and then ultimately molecules that are activated at signaling complexes, near the membrane, those molecules can diffuse into, for example, the nucleus. And we have very, very large endosomes that are represented topologically here. So EM represents endosomal membrane, and EN is the endosome, the interior of the endosome. NM is the nuclear membrane, and NU is the nucleus. And the idea is that in each of these regions, there are neighborhood relationships. So molecules in the extracellular space can interact with molecules on the cell surface that are associated with the plasma membrane. Plasma membrane molecules can interact with molecules that are in the cytoplasm. Cytoplasmic molecules can interact with plasma membrane, also molecules that are on the surface of other organelles that are inside the cell, and so on and so forth. So these topological relationships define what things can react with what other things. And one way to bridge, again, a cellular model that's formed as a reaction network with a cellular model that's explicitly spatial is to define this this topology, and we can even give properties. We can associate properties with the compartments that affect the reactivity of molecules within the compartments that we could then transfer into our spatial models. And you'll see exactly how this pipeline works through some of the examples that will be presented tomorrow. And that will enable us to compare models that start as reaction networks where we have well mixed. Essentially, we're assuming that all the compartments are well mixed and that molecules are heterogeneous or homogeneously distributed in those compartments, going to where models of where the molecules are heterogeneous in space. They're point particles, and they can be non-uniform in their distribution in any of the compartments that's described. So so this is kind of we need a way to bridge these two pictures, and I'll describe basically the system that we're using to do that between BioNetGen and MCell, which is a nice, I think it's fairly intuitive once you get used to it, but as I said, it has a little learning curve associated with it. So one of the things that we need to talk about is units of reactions. So if we consider, so basically the reactions that we have to worry about as having particular units are bimolecular reactions. And the reason for that is explained here. So when we do a simulation, when we do a simulation of when we wanted the differential equation for any species, so it's going to have units of concentration over time or number over time, and we need to know there's a rate constant that's associated with the reaction, and multiplying either concentrations or numbers of molecules of each of the reacting species. And I've given a little calculation here. And damn it, all right. And so now I have to remember how this per molar per second. This is meant to be the units of K2 that I'm describing here. So in sort of standard chemistry units, both the concentrations would be expressed in molar quantities. So we have essentially molar squared here, and we want the result to be moles per unit time. So the units of this rate constant have to be in inverse molar and then 1/seconds. And so what's shown here is converting between inverse molar per second and 1/number per second, which is what we need to carry out a simulation in the discrete units that we've been that we said we prefer in BioNetGen, and when we describe molecular events at the individual particle level. So in order to do that conversion, we have to essentially divide by Avogadro's number times the volume of the compartment in which that reaction is occurring. So the upshot of this is that in order to convert a standard bimolecular rate constant, which would be in inverse molar, inverse second units, we need to divide by Avogadro's number times the volume of the compartment, V. Okay. So frequently, if we have rate constants that are in standard chemical units, in order to convert them to the number units we like to use, we're going to have to divide by Avogadro's number times the volume. Now, if you write a bimolecular reaction in MCell, which you'll do tomorrow, and you enter the rate constant in inverse molar, inverse seconds, MCell will automatically do the conversion necessary to make the reactions occur at the appropriate rate and with the appropriate probability. On the other hand, if you tell BioNetGen that the rate constant is in inverse molar, if you just write a rate constant that has units inverse molar, inverse seconds, and you use that to describe a reaction in which you've described the concentrations of the species in number units, BioNetGen will happily carry out an incorrect simulation for you. You have to do
Well,
the
and actually, it's not BioNetGen's fault.
well.
Actually,
It's not, I'm telling you.
it's the solver's fault. It's the CVOD solver's fault.
Well, I wouldn't know. The CVOD solver just knows
No,
the
it has,
function that you gave it. It doesn't
no,
assume.
it has numerical instability because of the dynamic range of values. It's a numerical precision problem, I think.
Well, there's two potential problems. But the basic problem is you're going to give it the wrong equations, because if you don't do the unit conversion first, the number will be completely out of whack. It will be incorrect it will give an incorrect rate because you haven't done the unit conversion. So all right, so how do we do this? So this is a little bit complicated. So what we know that normally we have to convert a rate constant that's in sort of standard chemistry units or intensive units here, inverse molar, inverse second, to something that we could use in a stochastic solver, or that BioNetGen can use. You have to divide by Avogadro's something related to Avogadro's number and the volume. Now, and so it also comes down to depending on what volume units we use. So the trick here is that it turns out, so if you're a chemical engineer, then a standard unit of volume would be a liter. But from a cell's point of view, that's a preposterous number. And so cells really have dimensions on the order of microns. And so we like to describe the lengths that are associated with cells in terms of micron units. And if we then go further and talk about other surface areas and volumes associated with cellular length scales, then we end up with microns squared and microns cubed. And microns cubed is the relevant quantity here because we're interested in volumes. So a micron cubed, you should know, is a femtoliter. So like a standard bacterial cell, which you could think of as a cubic micron, roughly, if you're a physicist, you like this, then is a femtoliter in volume. And that's a reasonable, if somebody says, what's the volume of a bacterial cell? It's on the order of femtoliters, right? And a very, very small cell might have less than that. And in a large there are bacteria that probably have volumes that are approaching picoliters. But a standard E. coli has a volume of about 2 femtoliters. So so we want to use the appropriate units. And so when we make, for example, when we define geometries in M-cell, the units that we typically use and that Cell Blender is going to use to make those geometries are micron units, and the volumes that it's going to come up with are cubic microns. So it's very convenient to work with volumes in cubic microns for BioNetGen models that we want to turn into M-cell models. And so that's what we're going to do in the examples that we have. And so if you have volume in femtoliters, then the appropriate Avogadro's number to use is 6.02 times 10 to the 8, because we got rid of 15 orders of magnitude by going from a liter to a femtoliter. So we'll see this 6.02 E8 conversion factor in some of the models that we encounter where we're dealing with spatial compartments that have units of femtoliter. And so I apologize for I don't know how articulately expressed that was. It's like it's late in the day and I've been talking for a long time, but I hope that if you take away nothing else, you should know that the appropriate unit of measure to use for a cell is a micron, and the appropriate volume unit associated with a micron is a femtoliter. And if you then express all of your cell volumes in femtoliters when you make any compartmental model, at least in BioNetGen, things will come out much better, and the solver will not give you crazy results. Okay, so the other tricky thing that's interesting is the idea that we can get away with thinking about all the compartments in a compartmental model as being three-dimensional in some sense, in the sense of the reaction volume. I'm glad people like this. I hope this is the thing that you're finding exciting. The reaction volume, essentially, when you have a membrane compartment that's in 2D, you have basically a very, very flat compartment that has it, but it still has a volume because it has a thickness associated with it. And so one way to think about the biochemistry that occurs in membranes is that it's the biochemistry that occurs in a volume restricted or dimensionally reduced compartment that has an effective volume that's much less than, for example, if you compare the plasma membrane to the cytoplasm, the effective volume for reaction is basically reduced by several hundredfold, and that's because the thickness of the membrane of about 0.01 is about 0.01 micron, so it's on the order of nanometers. 10 nanometers is what we typically think of, compared to the order of the diameter of a cell, which is again order microns. Could be 10 microns if we're a eukaryotic cell. I should have mentioned back there that typical eukaryotic cells have dimensions of tens of 10 microns, for example, and so the standard volume of a eukaryotic cell instead of a femtoliter is a picoliter, roughly. So it's a cube, a 10 micron cube has a volume of a picoliter. And okay, so if we compare reaction rates in a membrane compared to, so in the plasma membrane compared to reaction rates in the cytoplasm, the effective volume in which those reactions occur is typically, as I mentioned, about 100-fold different. And so reaction rates are accelerated by being confined when the reactants are confined to the plasma membrane. And so that concept forms the basis for how we model the effect of compartmental volume and topology on reaction rates in BioNetGen. So this is what we do to model to make a. All right, can I get out of? Can you guys see? I don't know if the I'm just moving the window so that it's not cut off here. Okay, so when we skipped over the compartmental specification earlier on when we were defining our first model, this is the kind of specification that we were skipping, and this is how compartments can be defined in a BioNetGen model. So there are a total of 4 columns in the specification, and the first one defines the name of the compartment. So on each compartment line, and you can get this in the VS Code snippets, each compartment line starts with the name of the compartment and the dimension of the number of dimensions of that compartment. It's really either 3 or 2. So if we start and we go from outside to inside, so if we're making a compartmental topology, why did I do that? We start extracellular, and we have that's a 3-dimensional. That's a 3-dimensional volume, and it has volume, whatever we're going to call it. Typically, sorry, I'm on a conference. Thanks. Right. So this will have whatever volume. As I said, this is typically, if we think about, it's really the extracellular space between adjacent cells. If we have a suspension, that could be, very large. It could be nanoliters even. So millions of cell volumes in total. But I don't know, what we typically use something like like a nanoliter for the extracellular volume if we're modeling cells in suspension. but it could be much smaller than that too. So when we have like an M cell model, we can model a much smaller extracellular volume. And it doesn't really matter the extracellular volume is not going to be a critical parameter, except in cases where we have very, very low concentrations. Yeah, the dynamics of compartments. So far, we cannot have, well, Tom can field that question in the context for M-cell, in the context of BioNetGen. So far, the volumes that we model here are fixed. We don't allow the volumes of the compartments to change in time. That's something that we can actually relatively easily address in the differential equations, but we don't do that at the moment. That's the simple answer. And yes, it's possible. It's always possible to do more. Okay, so dimensionality and then volume. And it's going to be interesting. So you need at least these 3 entries for every compartment in your model. So name, dimensionality, and a parameter that's giving the volume. This could be a number too, by the way. And this number is going to be, for practical purposes, we always want to give this number in femtoliters or cubic microns, because that makes it possible to convert the model fairly easily into M-cell, number 1, and number 2, it keeps us from having very strange numbers here. So, and doing things like what Tom was alluding to earlier, getting numerical underflows in your solver because you have teeny tiny volumes that are far below machine precision. So, okay. I see people are cleaning around my office, which is, I was just being distracted by that momentarily. It's like, let's just put it this way. It's late where you are, and it's late enough here that the people who clean the department have showed up, because working hours were supposed to have ended. Okay. All right. So the outermost compartment is what we define as the root of a tree. So you can see another representation of this topology is as a tree. And the root node is the extracellular compartment. It contains everything that's inside of it. It's inside of it. And then the first child is the and so, well, any 3D compartment can have an arbitrary number of children. So we can have embedded within a 3D compartment any number of 2D compartments. Do we have any? Yeah, okay. We'll see an example of that in a sec. So, so we start out here. The compartment that's the first one that's embedded within the extracellular membrane, and there's only one that's a direct child for the extracellular space. We'll get to the cytoplasm has multiple children. So all the 2D compartments are embedded within this extracellular, and then the 2D compartments also have an effective that work. Also have an effective volume, where the volume is given by a surface area, that's this SA, which could be expressed in square microns, times some effective width. So the effective, it should be really thickness, effective thickness of the membrane. And that gives us a quantity that's also expressed in cubic microns or femtoliters. That's going to be the effective volume in which the surface purely, reactions of where the reactants are confined to the surface are going to take place. And then the final entry now that's required for all compartments that are not at the root of the tree have to have a parent. So here the parent is the molecule, is the compartment that contains that compartment, and here it's the extracellular compartment that contains the plasma membrane. So obviously, when you're making such a model, it helps to have at the very minimum, this picture in your head. If you don't have that picture in your head, you can draw it on a piece of paper, or you can cheat and look in the BioNetGen. We have a cheat sheet that has compartmental modeling explained in it, so that you can remember how this works. And you can also use snippets that help a lot too. Although I haven't fully investigated those, we have to. There might be some debugging that we still have to do. But okay, so now we have our first surface compartment, and we see that each surface compartment can contain at most one volume compartment. And so the immediate child, each surface compartment can have at most one child. Yeah, I mentioned the cheat sheet without further, and I had meant to give people a short walkthrough. So everything, all the key ingredients for modeling, all the key references can be found on the bionetgen.org website. We're currently working on improving the organization here. An important transition for us is going from emphasizing rule bender to de-emphasizing rule bender, but we're not quite there yet. But the key place here are the references. So quick reference, the quick reference guide I mentioned is here, and the actions and arguments guide that I alluded to a little bit earlier as well, which provides all of the actions and the parameters that are associated with them. So both of these two things are nice to have handy when you're building models. Yeah, and yes, the references are also in the last. I put a description in here for exactly this purpose, because it's a lot to remember, and we want to make sure that people know where to find things, even if they can't remember all the details. So like a quick reference guide is here. There's a simple BioNetGen model. This is a little bit out of date. It doesn't have all the context highlighting in it, but it does show proper syntax, and the topology and the syntax of the compartments block is highlighted here. The semantics of compartments are not really well explained in this. I don't think the model has the compartments block in it. But there are examples that are on the BioNetGen tutorial site. And thank you for the reminder that when we get to the end of the slide presentation, the next couple of slides have information on reference. And we'll go through an example before the magic hour of 4 p.m. Pacific time. Okay, so we can go through this whole hierarchy, and the key thing is, the thing that I think is trickiest here is just to remember that you have to specify the parent to tell BioNetGen what to connect each child compartment to. So you're basically specifying the topology that's represented by this tree graph, directed graph, through this syntax. And then once you've done that, BioNetGen can now basically determine what things can react with what other things, depending on what compartments they're in and whether they're in an adjacent compartment that allows the reaction to occur or not. So, for example, if you have can I draw? Yeah, I can draw. Except I can't draw it with the laser pointer turned on. I can't. Ooh. Guys, PowerPoint just does really weird things. PowerPoint and Zoom just gets really weird after a while sometimes. All right, can I draw? Yeah. What did I want to draw? That's a good question. No. Oh, I can't draw. And now I could. Oh, yes, I wanted to draw. And I've completely lost my pointer now. I put text. Nope. Hmm. I'm going to see. Yeah, I don't have a pointer on my screen. So it's hard to figure out what I can point to. If only these tools were as robust as the tools we're developing, we'd be in good shape. All right, I stopped the share for just a sec, and I'm going to try it again and see if I can. Let me just really quick go back to PowerPoint. All right, things are back. Let's see. We can try this again. All right, I got my pointer back. I'm not going to be so greedy as to try to draw. Suffice it to say that if we have reactants that are confined to one compartment or another, so if we have receptors that are in the membrane that can dimerize, then the reaction will occur in the membrane, and BioNetGen will automatically use the volume of the membrane to scale the reaction if we have a compartmental model. And similarly, if we have a bimolecular reaction and both reactants are in any compartment, we use the appropriate volume. What things become interesting when we have reactants in adjacent compartments. So if we, for example, have a ligand molecule that's out here in the extracellular space and a receptor molecule that's on the surface, what's the appropriate volume to consider when determining the rate at which these two things will interact? And the answer is the appropriate volume to consider is the three-dimensional volume in which the ligand molecule, let's call it L, will diffuse. So BioNetGen will assume that the rate of reaction is governed by a mass action rate law that's the product of the ligand and the receptor numbers and a rate constant that's scaled by the volume of the extracellular space. And so it's only when both reactants are surface bound that we're going to use one of the surface volume compartments to carry out a reaction. Similar thing will happen if you have a molecule that's within the cytoplasm that's reacting with a receptor at the surface. Again, the appropriate volume that will define the rate of the reaction occurring is the three-dimensional cytoplasmic volume. And so really again, this just goes back to this idea that enhancement of surface reactions happens when both molecules are confined to the surface. And it's very interesting how many there are many, most of the molecules that are involved in signal transduction have multiple means by which they are recruited to the plasma membrane in order to interact with various membrane-bound proteins, whether they are surface receptors, transmembrane adapter proteins, or ion channels of various forms. So there's always this tendency to nucleate. Oh, cursor gone again. Very, very exciting.
I'll just interject one thing here.
Yeah, go for it, because I'm trying to find my cursor.
Yeah, small conversation going on in the chat. So what Jim is talking about for the accessibility of molecules in the volume to access receptors on the surface is true if you think of the volume and the surface molecules as being well mixed and equally accessible from all points in space in the volume, right? And if it's well mixed, then it's like a point. Each of these compartments is really like a point compartment, and everything is equally accessible from all points. So it's only so this is this is only true in the well mixed case, and we'll see examples tomorrow when we start using MCELT, where if you set things up so that things are well mixed and kind of stay well mixed, then you'll get results that will agree with what you see in BioNetGen. But as soon as you violate this well mixed assumption, then you'll get a departure from those results.
Right. Right. All very good points. Thank you for bringing them up, Tom. Yeah. So yes, this is an approximate treatment of reaction diffusion. And if we want an exact treatment of reaction and diffusion, we had, better either use partial differential equations, but which again represent a continuum approximation, or particle-based stochastic methods, which is what MCEL is based on, that are going to account for the fact that things are not well mixed, that there's heterogeneity in the distribution of the particles within the cytoplasm, and particularly within the plasma membrane, and that those spatial effects can affect reaction rates. So if you want an exact treatment, you have to use a more exact method. And the whole, really the principle here that we're advocating is that we want to be able to make a model that starts at a fairly simple abstract level, and where we make certain approximations, but we should then go deeper and try to test whether those approximations are reasonable for the dynamics that we're considering. And for many kinds of things that take place in in a synaptic region, it's not reasonable to treat things as well mixed, and it's not reasonable to treat the particles as continuum. We really need to consider that they're localized and heterogeneous. But we got to start somewhere, and we're building up a set of tools that allow us to test different approaches. Okay, that's the end of the editorial comment there. I think we're mostly to the end of the prelude. Okay, at least I was able to change the slide. And we'll stop. And it's when I try to do annotation in Zoom that PowerPoint and Zoom stop playing nicely together. And they does things like make my pointer disappear. Oh, it just did it again. Oh, but it came back. Okay. And that is troublesome. Okay, yeah, so more sources of information about compartmental modeling. I mentioned the website. The tutorial has some really good starter examples about compartmental modeling, just very simple compartmental models with just a couple of components and a couple of compartments. The link is here. This is what it looks like. The ligand receptor binding models are a good place to start. The compartmental versions are labeled as shown here. And so you can see, how we model something using, the non-compartmental version, and then making a compartmental version. And what's nice about the way that we set up, we sort of went to the trouble of setting up this compartmental topology in order to minimize the amount of change you have to make to a standard model in BioNetGen to make it compartmental. So typically what happens, for these ligand receptor models, we don't actually modify the rules at all. We only modify the initial placement of species in the model. And I'll show an example. We still have a few minutes left. We'll quickly have a look at some of these simple models, and you'll have an example. You'll have an opportunity to see more in the coming days. So the tutorial tutorials definitely the place to start. The quick reference guide, as I mentioned, has a handy overview of the syntax and the pictures that I showed. Of course, the slides that you have now also have that, and actions and arguments that I already mentioned as well, which are available on the website. And I did not advance the slide. Thank you very much. They have a full documentation of the action syntax. Yeah, and someday in the future, these links will change. And so just, check us out on BioNetGen as all of this documentation evolves. Yeah. Okay. And I see a couple of questions. When will the recordings be made available? I think last year there was some fairly lengthy delay. I think Rosita has something to say about that.
Yes, yes, yeah. This year it will be pretty fast. I can say a couple of weeks. Yeah, hopefully, because we have another. We recently had our hands-on workshop. We just finished, and we have to clean up that recording first, release that one, and then this one. So I hope, yeah, you can wait for a couple of weeks. But yeah,
Is it not possible to share the links that come off of
it is
Zoom?
possible. Yeah, yeah.
Because we don't need to edit them. I really don't. I mean, I feel like people can just go through it as they want to.
If you want, we can do that. Sure.
Yeah, I would prefer that just to, because usually it's, I don't know how many hours Zoom typically takes to return those links, but we could post the preliminary versions of them as soon as they're available, as far as I'm concerned.
Yeah, yeah. I actually downloaded all three Zoom sessions for today, but yeah, there are like a huge part of it which is just empty, you know. But if you want, I can share that as is, and then we clean them up later, and then share them in our YouTube as a cleaner version. Sure, I can share them with everyone.
Great, thanks. I really appreciate that, because I think I think it's good for people to be able to review the material, close to the time that they first hear it, because that as time goes by, everyone gets busy and we'll have less time to go
Sure,
back.
sure.
Look at
And I
that.
will make sure that you have them pretty fast.
Great, thanks. I really appreciate that. Thank you.
Sure, sure. No problem.
So this is actually the last slide in this presentation. We can go through. Let me just pull up. I'll share the screen again and pull up the tutorial, the tutorial website here. It should be here, and we can look at a model or so. So if we scroll down, so tons of examples of rule-based models, which we only got to some of today, but we can do more on Thursday at the minimum with some of these models. And I would encourage you to go through some of the exercises that I put in the slides if you want to get more practice with stuff that we did today and with the concepts. And then all of the tutorial examples will be great to try out in VS Code. If people hit issues like they get syntax errors and things from some of the models, please let us know, because they haven't all been modified to run in the VS Code extension. So there might be little glitches you hit. I don't know. I don't expect too many, but there might be a few things. So if we just look, let's just look briefly at a simple ligand receptor binding model. And just to facilitate that, let's copy the text and make a model in the, what do I have? In the VS Code, we can make I get confused with what's a screenshot and what's the real thing. And I'm in the source control view, and oh, this is, let's not do it here. Did I close? I guess I closed the one we were working in earlier. Let me just do that real quick. Just working in a simple space. Yeah, day one here. Because that's not version controlled, and that will make me less. Don't worry about that. Let's just so we can make a new file. And copy the code in there. And we see that the syntax highlighted recognizes it. And we see the model. So we have the parameters here, this Avogadro's number times V. Well, what we call this NA capital V, this is the conversion factor that goes from molar to number per cubic micron. So yeah, right. So this NAV, this is what we use to convert, that we have to divide by to convert a bimolecular rate constant in per molar per second to cubic microns per second. So right, this is slightly confusing, this conversion, because when you can I guess you convert molar to inverse cubic microns, but you typically convert per molar to cubic microns. So that's anyway confusing at the end of the day. We have our here I made a mammalian cell. It has a volume of 1,000 cubic microns. We actually are only concerned about the surface, so we don't use the cell volume. The extracellular space is we made it 1,000 times larger than the cell volume, so it's going to be a peak. Is that a peak? Is that end up being a nanoliter? Something? Yes. And the plasma membrane thickness is set to be 0.01 microns.
Picoliter.
Did I say?
You said nano. I think it's pico.
Well, this is 1,000 microns cubed, so that's a picoliter, and then VEC is 1,000 times V cell. So
okay.
That's a nanoliter, right? I can still do that at the end.
Yeah.
It's a million times, yeah. This is pretty typical, like I said, for cells in suspension. They have a density of like typically on the order of something per mil. What's the hell? 10 to the 6 per milliliter. 10 to the 6 cells per milliliter is a nanoliter volume. Okay. Okay. So the area of the cell is we said it's 1,000 square microns, and then so that the volume of the plasma membrane is then, on the order of 10 femtoliters, 10 units.
And I want to point out, you have to be a little bit careful with what volumes and surface areas you use here, because you can make something that's physically impossible. Yeah. Right.
Yeah.
You can't have a surface area to volume ratio less than 0.66, right? Because that's the surface area to volume ratio of a sphere. Now I'm just
yeah. No,
that's I didn't do the math here, but I'm just pointing this out. Right.
Typically, the best thing to do is to pick a radius, and then use that for your volume, and then the surface of that volume, you can make the surface area of this, so 4 pi r squared times
right.
That's safe. That's usually what to do. It's
that's safe, and you can make the area larger than that if you want, but you can't make it smaller than that.
Right. It's tricky when you have something like endosomes where they're really many. So obviously you can make the surface area much larger, but there are really many of them, each of which has a
right. Yeah.
A small radius.
Yeah, but I was talking about per element, right? Per individual
kind of
element.
Fudge that one a little bit.
Yeah, you can fudge that.
So anyway, all of these quantities have been defined such that the volumes are in cubic microns, the areas are in square microns, but we convert, and then the numbers, the initial ligand concentration and receptor concentration are in number units. So the number of ligand molecules is the concentration in molar here times this conversion factor for to go from molar to cubic microns, and then or inverse cubic microns, and then times the volume of the extracellular space. So because the number is the concentration times the volume, and so we can see that the size of the space determines the effective number of ligand molecules that we're simulating. And sometimes it can be kind of excessive. So if you actually were to do a stochastic simulation here, the number of ligand molecules could easily be tens or hundreds of millions, and it's kind of silly to do that. So it's typically a good idea to reduce the volume but leave the concentration the same. Okay, and then the number of receptors here we picked as a 10 to the 4, and we see so here we only have one binding interaction that's got a forward rate constant Kp1. That's a bimolecular rate constant, and a reverse rate constant Km1. So we don't have to convert first order rate constants because they just have units of inverse time. But for forward rate constants, if we give it in per molar per second, we have to divide by our conversion factor to get it in microns cubed per second. And we would do that for every bimolecular rate constant we have in the system. If we express it in molar unit per molar units, then we can convert it to micron cube units, which is what we need for compartmental bioenzyme. And then we just have a very simple topology: extracellular, plasma membrane, and cytoplasm, going from outer to inner. We don't have any multi-child compartments here. And in fact, we don't even use the cytoplasm. We just have the extracellular and plasma membrane. The molecules are just ligand and receptor. And I'll just go down. So the only reaction we consider in the model is just ligand receptor binding. And notice that here we don't refer to the compartments at all. This is just, ligand and receptor can bind to each other. And then what compartments they happen to be in, they have to be in adjacent compartments or in the same compartment for this reaction to take place. And furthermore, the rate is going to be scaled by the appropriate volume. So that when we define a compartmental model in BioNetGen, the bimolecular rate constants need to be in these micron cube per second, and then BioNetGen will automatically apply the appropriate reaction volume by determining what volumes the reactions are present in, and what the appropriate reaction volume to use is. So that's the key trick. And then and it also minimizes the amount of work we have to do in thinking about surface reactions versus volume reactions and so forth. And then what reaction volumes get used depends on where we put the different molecules. So here, the ligand molecules are put in the extracellular space. And what's worth to note is that even when a ligand molecule binds to a receptor molecule to form a complex, that complex, the complex will now, the ligand is binding to the receptor on the surface, so the complex will reside in the plasma membrane at the cell surface. But the ligand itself remembers, and the compartment that the ligand molecule itself is in isn't changed by this reaction occurring. In order to do that, we would have to explicitly change the compartment of the ligand. So this complex exists at the plasma membrane, but the ligand remembers that it came from the extracellular compartment. And so if we dissociate the ligand receptor complex, the ligand will go back sort of automatically to the extracellular compartment that it came from. So in order to get ligand molecules inside the cell, for example, if we wanted to have a transporter, which we will have, we have to explicitly specify a reaction in which the ligand ends up on the intracellular side, so in which the ligand molecule explicitly goes to the cytoplasm. So since that's not normally part of receptor signaling, we don't do that in this model. But in transport models, we will have an example. And there is an example, the organelle model that's in the repo on GitHub. Where is it? It's also on another window in my VS Code here. If we go to this organelle transport model, we do have examples of reactions which transport molecules to different compartments. So I can't, how does this one go? So here, we're explicitly referring to the compartments of all of the species that are reacting. There's a transporter complex that's at one of the organelle membranes. And then when the complex breaks up, the transporter stays in the membrane, but this cargo molecule, which was originally in the cytoplasm here, goes to the organelle 1 volume, which is the interior. Okay, so this is an example of an explicit transport rule that can be specified in BioNetGen. In this model, we used unstructured species like we did in the first example, and we can use the compartmental syntax with them in a very simple way. You could also reformulate this using structured molecules. It gets a little bit more involved. Okay, so oops. So the other key thing that we have to do, as I mentioned, is we have to put, when we initially place the molecules in our system, so in our species block or seed species block, we can't just say that they're ligand molecules. We have to say that where the ligand molecules are, what compartment do they start out in. And here we've specified that they're in the extracellular compartment, which we specify using this syntax. We can also, as it's written in this other model, append the compartment name. This is a little bit of an unfortunate complexity in the syntax. When the compartment specification follows, comes after a molecule, the compartment is referring only to the compartment of that specific molecule. And when it's so in a multi-component complex, you can actually have molecules that are in different compartments as long as they're adjacent. As we saw, we could have a surface complex where the receptor is in the membrane and molecules can be in the cytoplasm or in the extracellular space that are bound to that receptor. When we have unstructured species, they can only exist in one compartment, as we have in this model. And so don't worry, I know this is confusing. It's the end of a long day, and I hope you haven't all tuned out, but we will get repetition to see these examples of these things again. So hopefully, you're building up a repository of knowledge. Okay, we're training your brain is what we're doing. So we have to place our species in the system. If I have only one choice about which syntax to use, I would use this syntax that's shown here, because it's a little bit more general. And it's weird, it's light outside, but it's pouring rain. It's almost like it's like sunny in the place that I can see, or not very, there's no rain clouds anyway, but it's pouring. Okay, so the receptor is put in the plasma membrane, and none of the rules refer to compartmental attributes, so they don't change this. The only thing that can happen is that complexes can form where, for example, the ligand molecule becomes attached to a receptor that's in the plasma membrane. And in order to demonstrate that, this model has a test observable in which we're asking the question, are there any molecules in which there's a ligand molecule that's bound to a receptor? That's what this pattern means, but the whole complex is extracellular. So can a ligand molecule rip a receptor out of the membrane? And the answer should be no, if we specified the model correctly and if BioNetGen is working correctly. And in fact, that's what we'll see if we run this simulation, which I could just do without thinking, and I have to, okay, yeah, it pulled up the right GDAT. And we can see that this test is always 0. So, no, you know, ligand molecules can't be bound, a receptor molecule can't be bound to a ligand molecule and have it be extracellular. It's only when they're at the membrane. So all these bound complexes are at the membrane. And we see that in this case, the ligand and receptor binding is set, the ligand concentration is set such that half the receptors get bound. And that's about it for this. I think that's most of the information that's in this compartmental example. We have some examples of more complex observables that are specified using patterns here. They're not really very exciting as observables in rule-based models go, but they are, you know, these are actual patterns. So what this pattern means is we define an observable called bound, in which we have a ligand molecule that's bound to a receptor molecule in the same complex. And that's what this, and then we want all the species that are specified by this observable. And what we can see, we didn't look at the net file previously, but if we just have a little glance into our net file, what we can see is that this reaction, the ligand receptor binding generates exactly one new species, which is this plasma membrane complex, in which a ligand is bound to a receptor molecule. And the complex is such, this ligand molecule, the reaction that created this complex didn't change the compartment of the ligand molecule. So it still remembers that it's extracellular. So it's extracellular side of the plasma membrane. The species description doesn't give the compartment of the receptor because it's in the same compartment as the whole species, as the plasma membrane. So it's a slight abbreviation. The receptor is actually in the plasma membrane as well. And what I was trying to get to is that when, so, you know, when network generation happens, the species block gets expanded to include all the species that are generated by the process of network generation. Here it's just one. And also the observables get resolved into what are called groups. So the groups are they define the set of species that summed over to compute the observable. And so what we can see is that there's the free receptor observable is comprised of just one species. It's just the free receptors that are not bound to ligand. The bound receptor observable corresponds to just the species in which the receptor is bound to the ligand. And this test observable, which asked if there are any species in which a ligand molecule is bound to a receptor molecule and the whole thing is still in the plasma membrane, that's basically a contradiction. There's no, that can't occur. And so there are no examples of that. So that species, that group is empty. And the reactions that were generated through network generation are listed in the reactions block. And we can see there was a unit conversion that happened, or a conversion of this, the intensive rate constant KP1 that was in volume per second. This 10 to the minus 6 factor represents dividing by the extracellular volume, which is a million, which has a value of 1,000,000 cubic microns in this case, because it's a nanoliter. Okay. And that is a simple as simple an explanation of the compartmental ligand receptor binding model as I can give. And I think we're well past time, so I don't know how many people are still left. But if you missed it, I guess you caught the recording.
Yeah.
I'm gonna stop there, and we still have 30 people. That's great.
Yeah.
Yeah.
Thank you very much.
That was a
And,
full day.
Yes, it
sure.
was.
Thank you too. I just shared 3 sessions with you. 4th is still running. So as soon as we end this session, not as soon, I guess maybe in 10 minutes or so, I will have the 4th one, and then I will share that one with everyone. I put them all in the general channel. So later on, I will cut it and then share it in YouTube. So yeah.
Right. We can also we'll put the links up on the schedule too, I guess. We get
Yeah,
it.
that would be good to put
Yeah, that's a good idea. Let me do that.
there.
Thanks.
Sure.
Good to hear people got VS Code working. One person.
Yeah.
It's really a great. I think if you get nothing else out of today, if you haven't used VS Code before, that's a good take home too. It's really a great tool. It's a great way to integrate all kinds of different projects that you might and that involve coding that you might work on. So
Yeah, I'm waiting for someone to come up with a way to customize the code editor that's built into Blender and customize it so that you can use an external editor like VS Code.
got it.
But I'm
Yeah.
not sure that's possible yet. But I saw discussions on the internet about people wanting to do that.
Is
so.
the code accessible in a file tree somewhere? Because you could, I mean, if it's just a file, you can definitely edit it from whatever you like, right?
Well, so the thing is, the cool thing about Blender is that your model can be completely encapsulated inside one blend file, meaning all the code and everything associated with that project is all together in one
Right.
blend file, so that when you share a model with someone, you share one file
Right.
and they have everything. But so what you'd want to do, and so if your code is living inside there, which it is, how would you edit it? And can VS Code be tricked into editing something in there, or what kind of abstraction layer can they put in between to make it so that your code inside the blend file looks like a directory on the file system or something, that VS Code doesn't have to know or care that it's editing something inside the blend file. And I think they'll be able to come up with a way to do that. Haven't seen it yet though.
Yeah, it's almost like it's a file system, right?
Yeah, and it is kind of a file system. So Blender itself makes a blend file look like a file system. So if inside Blender, for example, you say, I want to append something from another blend file, you can point at that other blend file from within Blender, and it looks like a file system. You can navigate through it. You can navigate through all the different parts of the model as if it's a file system. So all they need to do is trick VS Code into having that same view of the blend file. That would be cool.
I'm sure you could write an extension for that. I don't know.
Yeah.
You didn't look. Did you look in VS in the extensions?
I haven't looked yet.
Blender Python code templates, VS Code Blender Python. Some of the description is in Chinese. I can't, or in Chinese characters anyway. I can't read it. Tools to simplify Blender development. I don't know.
Yeah. But the obvious thing, right? To me, sounds like the super obvious thing. And I haven't seen it yet. But it'll happen.
You should check out, if you just Google, if you go into the marketplace and look for, and just type Blender, not a lot, but a couple of things pop up. I don't know if whether they address the issue that they look fairly nascent. Although this one has 16,000 They both have, you know, greater than 10,000 order 20,000 downloads. So that's pretty significant. So it might be worth seeing if they're useful.
Yeah, I'll check that out.
Using Microsoft Visual as external IDE for writing Blender scripts. So there's a link Michael found also.
Right, okay.
Yeah. Cool. Okay.
Yeah,
I don't have the brain power to look through that today, but it looks
yeah, I'm pretty burned out too.
Wait till tomorrow, Tom.
I know. I'm going to do another run through of what I'm going to show tomorrow, tonight before I go to bed. So just to have it all right there for tomorrow.
Yeah, it's definitely. I had the run through of making all those VS Code slides because I hadn't done that.
Yeah, those were really great, Jim. That was really fantastic. To have that.
Yeah, what's scary is that if we change, you know, the interface is under active development, and if we change it, when we change it, we're gonna have to at some point fairly soon have to remake all those slides.
Yeah.
That is, I mean, I got pretty fast at it, but still, that was a time-consuming process. That was more time and than I care to spend on making slides in a regular situation. But okay, well, you guys have been great. The people, and especially we were down to the very last members of the audience. But thank you very much for hanging in and for lots of interaction over the course of the day. And we'll see you all back again at various times, relative times.
Thanks, everybody.
Take care.
Thank you, everybody. And I also included all the links.
Thanks for all of that you've been doing all day long, Rosita.
Oh, sure. Thank you.
Don, I think, finally had to go to dinner. He's been awesome today. Him again and again. And Tom, as always, it's been great to have your input. And we'll see you tomorrow. Tomorrow I will add comments. I'm looking forward to tomorrow. I actually have to do the download, get the MCell 4. I still haven't done mine yet, so I'm looking forward to that.
Okay. It's gonna work. It's gonna work, Ryan, for sure.
I believe you.
As Bullwinkle would say.
Have you had a lot of trouble so far?
No, not at all.
I feel like our thing may be a little less stable. Just because there's so many different ways you can have the, VS Code, different platforms, and then just different Python and all that, it can get very messy, apparently.
Yeah. So we ran into a glitch earlier pointed out by JN and myself. We both had the same issue with older version of Mac running High Sierra. The BioNetGen Python module wouldn't run. It was built with the wrong library, and it was built with a newer library. So Sinan very quickly built one on an older Mac and showed JN and I both how to install it, and we got it to work.
He has a VM that for Mac now too. I don't know if he used that. He must have used some.
Yeah, I think he must have. Yeah, and he got it. Yeah, and it worked. So, and it'll be interesting to see, because I told him that when we build MCell, we build it for High Sierra, I think, right? Or El Capitan.
High Sierra. Yes.
High Sierra, and it works on all platforms. So you don't have to build it on more than one platform. You can build it on the older platform, and it'll work on the newer platforms. So that might simplify things.
Oh, yeah. I don't know. Until the Python doesn't work.
Say what?
I said
Well-
until the Python doesn't work.
Well, then, yeah, then I don't know what you do there, right?
Okay.
I think this is why Blender has a built-in Python.
Mhm.
I think that was one of the reasons why they distribute Blender that way.
Yep.
Yeah.
All right, Tom. What we need is Rosita. It's best, it's great. One of the best things you did all day was you say the session is over and you end it, and then it's gone and we stop talking.
No, I guess it was wrong.
Oh, no, it was good. It was really good. Yeah, but we have to, and keeping to time is very helpful. So
Yeah.
Okay, if you want, I can.
now I know better.
Didn't want to interrupt you. I thought that everyone is just asking questions, so I thought, okay, let's them keep going.
When things are a little more stable next year, I think next year I'll have the timing down better for that. I think I said this last year, but then we changed everything. So
Yeah.
it was very hard to predict how long it was going to take to get everyone through all that stuff. So now I know.
I think we did a good job of planning on our feet. On the
Thank
fly.
you so much. Thank you so much. From a participant point of view, feedback. Hi, Tom. Hi, Jim. Hi, Rosita. It was very, I love how it improves past 4 years because I attended maybe 4 years ago at CMU, 2017, if you remember, Jim. So I see the focus is that people understand. So the pace is not very fast and your focus is to be interactive and students ask questions in the chat and then there is a response for their questions, and also the pace of the instructors is not high. So it's word by word. And this is how it should be in the workshop. If we fast just go through the topics, people cannot follow and cannot implement in the Python or cannot install. They can just be the listener to the workshop and then later they can install and work. But now with the pace you have, they can do live, going to the folders, going here. Thank you so much. Yeah, I love seeing this huge improvement from the audience.
Thank you very much for a
Thanks
great
for that
comment.
feedback.
Yeah, and by any chance, Tom, do you have 5 minutes time now?
Sure.
Or-
Yeah, let's meet. I'll sign off here and we can meet on my Zoom channel. I'll start it up.
Okay. Thank you so much.
Okay, thank you very much,
Okay.
everyone. Have
Thank
a
you
great
so much, everybody.
Bye-bye.
Okay.
Bye-bye.
Bye.
Bye.
I'm going
Bye.
to close this session.
Okay.
Bye-bye.